Okay, let's wrap up our discussion of lipids by discussing how lipids interact with proteins. So proteins interact with the lipid bilayer. We'll call them membrane proteins. And we need to be talking about this because they are a major component of the lipid bilayer, up to 50% by mass. And these proteins can have all sorts of purposes. They can do chemistry if they're enzymatic. They can serve as channels and let ions or molecules go in and out of cells. They can perform signaling roles and much more. There are three main types of membrane protein that we'll discuss: integral membrane proteins, lipidated or lipid-anchored membrane proteins, and peripheral proteins. Let's start with integral membrane proteins. These go all the way through the membrane, so they start on one side and end on the other side of the membrane. These bind lipids super tightly. They are really stuck in that lipid bilayer. In order to get them out, we need to use something like a detergent to kind of suck it out of the lipid bilayer, or denature that protein to get it out, so change its shape. You might have heard these proteins called transmembrane proteins. It's the same thing as an integral membrane protein. Now, something interesting about integral membrane proteins is that all known transmembrane segments contain secondary structure, specifically alpha helices or beta sheets. So not turns and loops, just alpha helices or beta sheets. Why is this? Well, if we think back to the structure of peptides, you'll recall that the backbone, this is just a little quick dipeptide for us to talk about, that the backbone contains polar elements, right? We have the carbonyl functional group and the amine functional group. So these are rather polar aspects of this peptide backbone. But if you think back to when we first learned about secondary structure, you'll recall that alpha helices and beta sheets use these polar regions of the backbone to perform hydrogen bonding, which strengthens and stabilizes their structure. So what's happening there when these polar regions of the backbone do hydrogen bonding with each other, it makes those polar regions interacting with each other kind of hidden from the non-polar lipids of a lipid bilayer. Let's kind of visualize this for a second, right? So if this is my membrane, and I'm not going to draw too many lipids here, folks, so please just forgive the simplification. Here's my lipid bilayer. If I have an alpha helix going through that bilayer, that helical structure allows the polar aspects of the backbone to hydrogen bond with each other. And if they interact with each other, they're therefore not interacting with the non-polar bits of those lipid tails. So this is why all known transmembrane segments are containing either alpha helices or beta sheets. And so this gives us kind of a second way to use these hydropathy plots that were first introduced way back when we were learning protein structure. In this case, we're going to use any segment of the hydropathy plot that is in the hydrophobic region and assume that this region crosses the membrane, right? So remember that anything above 0 is nonpolar or hydrophobic, and anything below 0 is hydrophilic. So if this were a hydropathy plot for an integral membrane protein, we would assume that this region, marked as 1, would be a transmembrane segment because all of the R groups coming out of this segment need to be nonpolar in order to interact with the nonpolar lipid tails in the phospholipids or of cholesterol. Let's take a look at a few examples of this. So for example, there's this protein structure shown to you on the right called bacteriorhodopsin, and it is all alpha helices. You'll see that each of these alpha helices pictured on the right corresponds to one segment of this protein that is on the hydrophobic part of this hydropathy plot. You can literally count all 7 alpha helical sections. Each of these sections we assume must be nonpolar because remember, these R groups are sticking out of the protein and therefore would be interacting with any lipids in the lipid bilayer. Similarly, there can be structures that don't contain these types of alpha helices and rather are primarily composed of beta sheets. Similar story here, folks. The non-polar R groups are facing the outside, so you could look at all of the peaks in a hydropathy plot and say, oh, this nonpolar region probably corresponding to this beta sheet, and this next one probably the next beta sheet, etc. So let's relate this back to something we already know about protein stability. So previously we learned that protein folding is controlled by the hydrophobic effect. All of the non-polar amino acids are sequestered to the inside of the protein to minimize their interaction with water and to increase the entropy of water. How can we understand this when we're talking about a transmembrane protein that goes through a very non-polar environment? We'll have to think a little bit different about these proteins. We would assume that in terms of normal protein folding, where the core is nonpolar, the same should likely be true for a transmembrane protein. Still, if it's folding and hiding away the inside from water, that should be nonpolar due to the hydrophobic effect. However, the second bullet point is super interesting, that on average, the residues in the alpha helices or beta sheets that are exposed to the lipid bilayer, those residues on average are more hydrophobic than residues found on the inside of the folded protein. Why is this? Well, if you think about any R group kind of sticking out and interacting with lipid tails of a phospholipid, we should be able to understand that through the hydrophobic effect, this interaction must be very nonpolar. Otherwise, the entropy of something, maybe the lipid tails, for example, would be greatly decreased in this scenario if the R group were polar. So because lipid tails are so polar, because lipid tails are so non-polar, this really forces any of the R groups interacting with them to be extremely non-polar as well. Whereas on the inside of the protein, if we had a polar R group kind of sticking out on the core, maybe it could perform a hydrogen bonding interaction with backbone carbonyl oxygen or something, for example. And that wraps up the first type of membrane protein. Up next, lipid-anchored proteins. So a lipid-anchored protein has a lipid tail covalently attached to it. So you take a lipid, form a covalent bond from it to an amino acid on the protein. Now this covalent bond can happen on a cysteine, a glycine, or on a cysteine again. Exactly where isn't important. But what we should understand is it results in a protein that now has a lipid kind of sticking off of it. And so again, according to the hydrophobic effect, we should assume that that protein then inserts itself into the membrane. This type of interaction will be weaker than an integral membrane protein, because it's really just the interaction of this single lipid with the lipid bilayer that's really getting it stuck there in the bilayer. The last type of membrane-associated protein are peripheral proteins. So these bind very loosely to the lipid bilayer. I like to think of them as just kind of sitting against the bilayer, maybe inserting themselves a little bit into the bilayer. And this image shows you some different ways that these proteins can interact with the bilayer. Maybe just, for example, an alpha helix is slightly embedded and interacting with the lipid heads or the phosphate groups. Maybe a turn or a loop is just dipping down into that bilayer a bit. Or maybe it sits right on top and we have some ionic or ion dipole interactions between our groups and the phosphate or X groups on these lipids. Now, since these are only interacted really at a peripheral level with the bilayer, this is the weakest type of interaction between a membrane protein and the bilayer. And this is kind of just to summarize the three types of proteins that we learned. Integral membrane proteins go all the way from one side to the other side of the bilayer. A lipidated protein has a covalently attached lipid that sticks into the bilayer. And finally, a peripheral protein where just a little bit of the protein will either be touching the membrane or embedded slightly. And that is it for lipids, folks. We talked about the 4 types of lipids. Then we transitioned into more structural, functional thinking. What are the shapes of lipids? What does that mean about the function it can perform? And then finally, we talked kind of bigger picture about the lipid bilayer and proteins that associate with it. I'll leave you with this beautiful, cute cat bilayer.